Hello everyone. So today we will be discussing the last part of this chapter, that is the ingestion by the cell. Okay. So how cells ingest foreign particles from outside to the inside. Okay. So today we will be learning about ingestion by the cell. Okay. So the process by which the cell ingests the foreign particles from outside to the inside, that process is known as the endocytosis. Endocytosis. Okay. By this process, the cell ingest. Okay, endocytosis. And this endocytosis is further of two types. One is the pinocytosis. Second is the phagocytosis. Okay. So endocytosis is the process by which cell ingest, and this endocytosis are of two types: pinocytosis and phagocytosis. So starting with pinocytosis, it is also known as cell sinking. That means smaller particles, the macromolecules, are being ingested. Okay. So what are macromolecules? Like proteins, some nutrients. Okay. So those small particles are being ingested by the cell via pinocytosis. Okay. Now, if we talk about phagocytosis, it is called as cell eating. That means, contrary to pinocytosis, larger particles like bacteria, based cells are ingested. Okay. So now you can get the difference between pinocytosis and phagocytosis. In phagocytosis, larger particles like bacteria, dead cells are being ingested, and in pinocytosis, smaller particles being ingested. Okay. Now, how pinocytosis actually takes place? So we will see step by step. Okay. Now, this is the outer surface of the cell membrane, and in this outer surface of the cell membrane, there present some receptors. Okay. And these receptors are being assembled to each other in such a way that it formed a coated pith. Okay. So this assembly of the receptor is called as coated pith. Okay. Fine. Now the molecules which are need to be ingested by the cell first need to get attached with this particular receptors. Okay. Suppose these are the molecules. Okay. So it need to get attached with this particular receptor. Fine. This is the membrane. These are the receptors. Okay. So these are receptor. These are macromolecule. Okay? So this macromolecule get attached with this particular receptor. Okay. After get attached with this particular receptor, beneath the surface of the membrane, there are some proteins. Two types of proteins generally present. Those are the lattice work of the fibrillar proteins and the contractile protein. Okay. The fibrillar protein is known as the lectin, and the contractile protein is the actin and myosin. Okay, fine. So when this macromolecule gets attached with the receptor, which activates this protein, and after that, what happens? Invagination occurs. So after that, what happens? By the action of these proteins, the invagination takes place. Okay. So these are the receptors. These are the macromolecules. Right. Now, what happens? Since this is the outer surface of the cell membrane, so in outer surface, what things occurs? That is the extracellular fluid. Okay. So in outer, there is extracellular fluid. So when invagination takes place, the extracellular fluid also will enter in this place. Fine. Now in extracellular fluid, we all know calcium ions are present. Okay. So this calcium ion will react with the actin and myosin that is present beneath the cell surface. Okay. So calcium that is present in extracellular fluid will react with actin and myosin. Okay. And which results into pinching of the invagination from the surface of the cell to form a pinocytotic vesicles. Okay. That means the invagination will pinch off from the surface of the cell to form a pinocytotic vesicles. This is pinocytotic vesicle. Okay. These are the receptors and these are the macromolecules which has been understood. Okay, fine. So clarithrine also can do the same work that breaks the invagination from the surface of the cell. But in here, the contractile protein, that is the actin and the myosin, plays the most predominant role. Okay. So by this way, the pinocytotic vesicles formed via invagination. And the clarithrine and actin and myosin helps. Okay, fine. Now, what happens? Phagocytosis is almost pretty much the same. Okay. But in this case, larger particles are being ingested, like bacteria, dead cells, etc. But in case of bacteria, in the process of phagocytosis, what happens? Suppose this is a bacteria. Okay. So in bacteria, there are some attached antibodies. Okay. some attached antibodies are present along with the bacteria. So this attached antibody makes more visible to get attract to that particular receptor and finally gets ingested into the cell. So this intermediation of this particular antibodies is known as the opsonization. Okay. So in phagocytosis, this opsonization process also takes place. Fine. Now what happened? After these pinocytotic vesicles form, okay, after this, we got pinocytotic vesicles where macromolecules are present. So we all know inside the cell, lysosomes are also present. So these are the lysosomes. Okay. Inside the lysosome, there is an enzyme known as acid hydrolase. Okay. Acid hydrolase. These are lysosomes. This is pinocytotic vesicles. Okay. Now what happened? This lysosome will get attached with these pinocytotic vesicles. After that, what happens? After being attached, this lysosome will transfer this acid hydrolase into the pinocytotic vesicles. Okay. These are the macromolecules and these are the enzymes which are needed to digest these macromolecules. Okay. So this acid hydrolase will digest this particular macromolecules and those digested parts are being effused out of the cell. Sorry, that's okay. And this particular vesicle where the lysosome has given the enzymes to this particular vesicle is known as digestive vesicle. Before it was pinocytotic vesicles where only the macromolecules were present, but after being attached with the lysosomes, when the lysosome transferred its enzyme, that is the acid hydrolase, to the pinocytotic vesicles, and it gets converted to digestive vesicles. Okay. And these digestive vesicles, where digestion takes place of these macromolecules, the digested part gets diffused out of the cell. Indigested part that part that is the present inside the cell will be removed via exocytome. Okay. That means like if this is the membrane of the cell, this particular digestive vesicle will get attached to this particular membrane, and from there the undigested part will get excreted. This is undigested part. Okay, fine. After being digested, the digested part gets diffused out of the cell, which is required for the nutritional purpose, and the undigested part gets excreted out of the cell via exocytosis. Okay, fine. That's all about the first unit, that is the cell and its components. So this was the last part. Today we have completed. Fine. So if you have any doubt, you can ask me in the comment box. And in the next video, we will be discussing the other unit. Okay. Thank you.